Hello, everybody. Today we're going to go over the reproductive system. It is there's one more chapter after this, and then we'll be done with the term. Okay, today let's get into it. It's only about 47 slides, so it's not the longest chapter. Okay, so the reason for the male and female reproductive organ is for reproduce and nurture of the sex cells, okay, and transport them into the site of fertilization. Male sex cells are called sperms and female sex cells are called eggs or oocyte. There's a special dividing method of dividing called meiosis. So it's similar to mitosis, but it goes even further, more dividing. So normally with the mitosis, we get 46 chromosomes, but with meiosis, we get 4 sets of sperm cells or egg cells with 23 chromosomes in each. Okay, and they're all different, so that it will have a better genetic varieties, genetic diversity for the next generation. And this is for diseases and all that to once you have a better diversity, it is less likely to have genetic problems. That's why it's not good to marry your brothers or sisters or close family members, because the you have less genetic diversities. And during meiosis, you might end up with the same thing, and then genetic disorder or familial disorders will be passed on. Okay, well, let's get to the anatomy of it. So this is a male reproductive system. Let's start with the scrotum. So on the outside of the scrotum, there's something called a raphe. Okay, it's just a raised thickness on the surface. Okay, there's 2 muscles here. The dartos muscles, it forms the pouch, and then it forms the scrotal septum. And then the cremastoid muscle, which is right here, is like a net surrounding the testicles. So these actually comes it's descended from the internal obliques of the abdominal wall, and it will cover the testes like a net. Okay, and it depends on the temperature. It goes up and down a little bit, and it's usually, the scrotum's usually 2 to 4 degrees Celsius below your core temperature. Because if it's too hot, it might denature the sperm or the protein, and then you have a problem. Okay. So here is a look at the testes, and then we're going to look at the epididymis and go from there. So in the testes, so here's the main body of the testes. On top of the testes, there's something called the epididymis. Okay, this is where your gonad, and which is your sperm, that's where it is developed. Okay, so wrapping the testicle, okay, there's something called the tunica vaginalis. Okay, and remember, this is the cremastoid muscle. It's like a net. And wrapping around the actual testicle, there's something called the tunica albuginea. Okay, albuginea, which divides these lobes. Okay, which form, these are the septum inside the actual testicles, which divide these lobes. Okay, 300 to 400 lobes with 1 to 4 seminiferous tubule per lobe. So usually around the 7th month, that's when the testicle descends. And when it doesn't descend, is a disease called a cryptorchidism. Okay, like orchidism. Okay, crypto. And that's when sometime 1 or 2 they don't descend from the abdomen. So we're going to look into these more into the seminiferous tubule. That's where the sperms is developed and released into the lumen. Okay, there are different types of cells. Okay, Sertoli or sustentacular cells. They secrete signaling molecule to for the sperm development and whether the sperm needs to be recycled. Whether it needs to live or die. Okay. They also form a blood testis barrier. Okay, it's like the blood-brain barrier. Also, it also helps keeps the germ cells and preventing them from escaping, so that you don't develop an autoimmune problem. Okay. Okay, and here's a Sertoli cells, Leydig cell. We're gonna look at that in a little bit. Okay, more Sertoli or sustentacular cells. So here's how sperm is formed. Okay, when the sperm is formed, it's called spermatogenesis. Of course, you're going to need testosterone, okay, or androgens, okay, for sperm to develop. So, begins at puberty, starting about 35 or so. Then it will start having a decline of the sperm production, and usually takes about 64 days for a sperm to develop, and a new cycle starts every 16 days or so. Okay, again, depending on the sustentacular cells or Sertoli cells. Okay, they're all the same thing. So here's what I talked about. The meiosis. Okay, so first it starts with mitosis. Okay, mitosis, you develop into two identical cells. Okay, then it goes into meiosis 1. Okay, meiosis 1. So it develops into two different kinds. And then meiosis 2, where this one divided into two different one, and this one is two different one, each one of these carrying 23 chromosomes. So this is what meiosis look like. Okay, of course this one as well will go back to here and then further divided into more. Okay, so meiosis will get the chromosome number correct. Okay, what about the structure? Spermiogenesis. So we're going to look at the sperm, what it looks like. So a sperm cell is 85,000 times smaller than an oocyte. So it's much smaller than an egg. Okay, if you guys ever seen a picture or a video of an egg being fertilized, you can see, the egg is like a giant sun, and then the sperm goes in, and then it will dig in. Okay, 100 to 300 million sperms are produced per day. Here is the what a sperm looks like. It's got three sections: the head, the midsection, and the tail. Okay, the tail is for swimming. It's called the flagellum. Okay, the midsection here, it's filled with mitochondria. Why? Because it needs power to move the flagellum. Okay, and here's the head. The head is got a covering, okay, called the acrosome, and is covering the head, which is where the nucleus is. As you can see here, once the sperm attaches to the egg and dig in, there's something called the acrosomal reaction, which I think we'll look at it in a minute. So now that the sperm is made, what to do? Okay, sperm will be transported through a tube, okay, called the seminiferous tubule. Okay, but it is then must go through the tubules and then go to the epididymis. Okay, then after it is then it will go into ejaculation. Okay, So the epididymis, as you recall, is on top of is this section right there, okay, or this section right there, the epididymis. It's another tightly coiled tubes, okay, connect to the duct within the testes, promote maturation of the sperm cells, and allow storage 12 days to move through. Here is where you have pseudostratified stereocilia for reabsorption. Sperm is motile when it leaves, so it is not movable until it leaves. Okay. Next, we have the ductus deferens. So here is the testes, epididymis, okay, and then it goes to the ductus, okay. That's where a lot of people they snip them so that sperm won't go through there anymore. But most people would call this the vas deferens. Vas deferens. Extend from the epididymis, move to the seminal cord, to the inguinal canal, and terminate in the ampulla right up here. Okay, and we're gonna learn that in a minute. Let me fix this real quick. Okay. So once the sperm is produced, it's motile, it's movable, and transported. How do we keep them alive once they leave? Okay, there's something called the seminal vesicles that will secrete alkalinic fluid because the female reproductive tract is acidic. It's got fructose for it to move, and prostaglandins to help with motility to keep it alive. Okay. Then the content will empty into the ejaculatory duct. Okay, and here's also the prostate will secrete another alkaline fluid, citrate, PSA enzyme that liquefy after ejaculation, and it helps it becomes less thick. Okay. So here you have the let's see, the vas deferens, right? We saw the vas deferens. Here is the ampulla. Okay. Here is the seminal vesicle that will release all these things. Okay. And then it's going to go into the ejaculatory duct and then out to the urethra. So also there is the prostate gland, which will also help release some of these things. So the sperm only makes up about 5% of the semen. Okay. And then once it is, first, it is quite thick. Okay. First, it's coagulate to help. Once it's ejaculated, it is quite thick and it helps maintain its shape, keep everybody together in the female reproductive tract. Then is this PSA that will then liquefy the semen so that it will for the sperm to go swim further. So the entire solution is called semen. The fluid the urethra convey to the outside during ejaculation consists of all these things. Okay, lots of nutrient for the mitochondria to work. The sperm cannot fertilize until something called the capacitation. So capacitation is when the sperm actually undergo a change. It has to go through something called the acrosomal reaction, like I told you about, and then where the the covering of the head of the sperm has to go through some kind of reaction, and then